So another concept that tends to be a sticking point for a lot of undergraduates is tonicity. So the thing about tonicity, so when we talk about isotonic, hypotonic, and hypertonic, well, if you know your prefixes, so isotonic referring to iso meaning same, hypo meaning low, hyper meaning above. So the tough thing about tonicity, it's like saying so it's a comparative term. So something isn't hypertonic by itself. You just can't just have one solution and say it's hypertonic. You have to say it's hypertonic relative to something else. So when we're talking about hypotonic and hypertonic in terms of medicine, we're talking about extracellular fluid relative toward the inside concentration of a solute inside of a cell, inside the intracellular fluid. So we're looking from the outside in versus the inside out. I know like there's a lot of people who like to create multiple choice questions where they say the inside of the cell is hypertonic or hypotonic. So whenever you're answering a question about tonicity, especially on these exams, especially if they're trying to throw you a curveball, determine which way are you looking. Are you looking from the outside in or the inside out? So the thing is that hypo refers to your starting point versus your ending point. So again, are you looking from the outside in? And typically in medicine, you're looking from the outside in. You're comparing the extracellular fluid toward the inside intracellular fluid. And why is that? Well, which can you change more readily? The tonicity or the not the tonicity, the concentration of your extracellular fluid or the concentration of your intracellular fluid. So which is easier to affect? Extracellular concentrations or intracellular concentrations? Well, to affect or to directly affect intracellular concentrations of a solution, you would have to inject a cell with a different solution to change its concentrations of a solute. Now, are you going to inject billions of tiny needles into every cell to regulate the intracellular concentration? No, right? That's really tough and then pretty much impossible. So what you're going to do is through an IV, maybe introduce another extracellular fluid and through osmosis try to change the fluid balance. So when we're talking about IV fluids, we're talking about looking on the outside in because when you introduce an IV fluid that is extracellular fluid, now you're trying to move water from extracellular fluid or actually between the extracellular fluid and the fluids inside of a cell. So you're looking from the outside in. Now, back to isotonic, hypotonic, or hypertonic. You're starting with the extracellular fluid, and then you have an intracellular fluid. Is the extracellular fluid less concentrated than the inside of a cell? It's hypotonic. If the extracellular fluid, again, your starting point, is more concentrated than the inside of the cell, it's more concentrated, therefore it's hypertonic. So again, when you're ever in medicine, you're typically looking from the outside in. Tonicity is referring to extracellular fluid, or a fluid that's being introduced compared to the normal solute concentration inside of a cell. And as you can see, by having different concentrations, if you have the same concentration, no net overall movement of water, therefore the cells stay relatively the same in this erythrocyte we see here, or red blood cell. Hypotonic solution, you have there's practically no solutes in this solution. So what's going to happen? Remember, osmosis attracts water toward the solutes. The solutes are like hungry undergrads or med students. They love all these sugars, they love all the salts, so they're attracted to it. So what we have here is all the only salts and solutes here, the sugary and salty parts, are inside the cell. There's no solutes out here. All the water molecules are going to rush in, and this is what causes that red blood cell seen in this electron microscope scan right here. This is why it's swollen up. Now, in the hypertonic solution, again, we're looking from the outside in. There's more concentrations of solutes in the extracellular fluid than inside of the cell. So because there are more yummy, salty, sugary snacks out here, the water molecules rush out of it. As they rush out of the cell, then the cell loses volume. And this is why we get that crenation, this kind of like loss of volume right here. Hypotonicity, again, it's just like have here using our hungry med student example. All the sugar here inside the cell, not so much sugar outside in this extracellular fluid. Again, we're looking on the outside in. Less concentration on the outside compared towards the inside. Therefore, is hypotonic. And which way are these hungry med students going to move? they're going to move where there's more sugar, right? So again, this is what happens in the hypotonic solution. And as more of them enter this crowded space, this, instead of being a rigid room, this is a flexible cell wall. So what's happening is as more water molecules pile into a cell, or say this is like one of those stretchy bounce houses or some sort of stretchy room. So as more people pile in, this starts to stretch and stretch. But if there's too many people in there, it can eventually burst, right? Same with the cell immersed in hypotonic solution. If you throw a red blood cell into a solution of pure distilled water, the water is just going to continually rush into the cell until it eventually pops. So this is what we call lysis. Now, hypertonic solution, so pickles, classic example. Pickle brine solution, very salty, has a lot of solutes. And are these as big as a regular cucumber? Notice that these pickles, they're pretty shriveled, they're pretty shrunk, they're not smooth like a plump like a cucumber. And why is that? Well, that salty solution drew out all that water from the inside of that cucumber. So this is the reason why it gets wrinkly and also shrinks as well. So same with the cells inside a hypertonic solution. As you have cells and then, so here we have a cell, but say you have a way more sugar or salt or something in the surrounding environment. This is going to draw out those water molecules. Again, if you have salts and sugars, they attract water through those polar or their or for in terms of ions due to their electric charges. So they attract water and draw it out of the cell. This causes the cell to collapse and lose volume. And this is what we call crenation.